All right. So, we have been talking about libraries, gene libraries, and last time we saw that, you can have either a genomic library or a cDNA library. Why do we even make libraries? See, gone are the times when people could freely share, microorganisms or any of the other tissue samples, freely across the world. That used to be the trend till maybe like, 80s or 90s. But then what happened was, people started, what should I say, kind of started misusing the national resources. So these are typically resources that are very very specific to certain countries. For example, again I tell you, this course is so beautifully integrated. In your next semester, when we talk about IPR, you will know that what biopiracy means, right? So typical example is, people in the 70s, took a lot of herbal samples from India for, testing it out in cancer cells. And, one of the drug it turns out that a common plant, a herb, was found to have very potent alkaloid, okay, which then was chemically synthesized. They made a patent and then they started making money out of it. Is there a harm in making money out of it? Probably not. But then, they took our resource and then, they started patent, and then, it looked as though everything was theirs. There is something that is there in nature. Nature has provided you. You just made the process efficient and then, got this patented. As I said, you will need a lot of things during your intellectual property rights management. How these things have become so complicated now that it is not possible to share microorganisms. Even if you find something novel, even when you communicate in any scientific journal, people's curiosity may be raised. So they may, they may like to get the samples and all that. You can't share samples, obviously. So what is the next best thing? The next best thing will be to share your libraries, gene libraries. Okay. As of now, this is unfettered. That means you could share your gene library across the world. You can get it from anybody. You can mail it to anybody. Legally, there is no bar in sharing these resources. Although, depending on what kind of a resource you are doing, shipping it out to somebody else or getting it from somebody else, you may have certain legal guidelines that you may need to follow. But by and large, Gene Library offers us a, easy way to share these resources. Okay. And gene libraries come in the form of both genomic library and cDNA library. And we definitely, I briefly mentioned in the last lecture, when it comes to genomic library, how do you even decide whether you want to have a genomic library or a cDNA library? By and large, the thumb rule is that when you're dealing with prokaryotes, the genomic library works beautiful because there are no introns, nothing. There are barely any, unnecessary thing. It's like an organism that is packed with all kinds of genes, right? So genomic library works really well for bacterial species, viruses, right? Prokaryotes in general. But when it comes to eukaryotes, you have an issue because the genome is probably only 2% of the genome codes, and rest of the 98% is up there, which is not going to code for anything. So you are going to be and unnecessarily wasting things with genomic library. Genomic libraries are interesting for certain reasons, but by and large, cDNA library is one that will be useful for a mammalian system unless you are trying to study the promoter structure or how a gene is being controlled, how the expression is being controlled, what kind of transcription factors are binding to promoter. Then you would need genomic library. Otherwise, for routine purposes, when you're trying to find a novel gene homolog, cDNA library works beautiful. Okay, so by and large, cDNA library when it comes to eukaryotes and genomic library for prokaryotes, you would definitely you will be using genomic library for understanding the promoter structure or how a gene gets regulated. For those reasons, we will be using the genomic library. Okay. All right. So now that we have a library, whether it is a genomic library or a cDNA library, the question is, how are you going to create this library? You know, in our class, we read that plasmids can carry a maximum. Theoretically it can carry about 15 kilobase pairs of foreign DNA, but in reality, not more than few kilobase pairs. Right? Maximum is about 4 kilobase pairs, beyond which plasmids tend to be unstable. They start rearranging, they start losing fragments of DNA. Okay, so it becomes difficult. Phages, bacteriophages on the other hand can routinely accept anywhere between 10 to 25 kilobase pairs, although in reality, people don't go beyond 15 kilobase pairs or 20 kilobase pairs. And then you have bacterial artificial chromosome, yeast artificial chromosome, and human artificial chromosomes, which can literally carry, in thousands and thousands of kilobase pairs. Okay. So by and large, plasmid and phages, especially created phages are used as cloning vectors. Are used as cloning vectors. In our beginning class, we talked about what cloning vectors are, what expression vectors are. So I'm not going to revisit. But once you clone, what is it that you're going to do, right? Let us just say you created a genomic library, and you may be interested in some gene. You want to look at, say, DNA polymerase from an organism, and somebody else sitting somewhere in the world, say somebody from Japan or US or UK, they would like to have a library from of your organism, and they want to screen for, say, a ligase and equivalent, or one of those enzymes that we saw in the molecular biology, this thing, say a phosphatase enzyme or something like that. So when they wanted it, cDNA library, they somehow you need to screen and figure out that this is the clone of interest. Okay. So as usual, I think I found a very nice this thing video from MIT, where Professor Eric Lander talks about how do we screen these things, right? So maybe we will start with that, and then we will go into what these libraries are, the types of libraries, their construction, and so we'll start the reverse way, because I thought this was a very nice lecture. So I think I would set the stage up for that. Let me share it. So I would like to apologize for this, for eating into your time, because I had a meeting, unscheduled meeting, so I had to go away. Otherwise, you would have had our class early, and you guys would have been free by now. I'm sorry that something like this happened today. Hopefully, it won't happen again. Okay, there we go. Is my screen visible?
Yes, sir.
Okay, thank you. So this is a 20-minute this thing. Then we will go back to our other lecture on the types of libraries, how they are made, how they are screened. Okay.
Good morning. So last time, we talked about the most remarkable biochemical purification procedure ever invented, cloning. You remember the issue with biochemistry.
You people should probably also go back and look at some of his videos. Okay, so there is this one called Basic Mechanisms of Cloning. Here, I think there are like series of videos from MIT open courseware. Some of these are so beautifully explained. So even after reading, to have additional understanding, you should probably go back and check these resources out. I mean, I've been telling this probably since last semester, I guess, or even before that, that MIT has these lectures that are recorded, and these are available. So you should utilize that. Okay.
You're going to grind up a cell. You're going to take the contents and run it over different kinds of separation columns or centrifuge it or things in order to separate some things from other things. The problem with purifying a gene that way, away from all the rest of the DNA in the cell, is that the gene has exactly the same biochemical properties as all the other genes. How in the world are you going to do it? The solution was devilishly simple and devilishly complex at the same time. All you have to do to purify something is dilute it. If I take any substance and I add enough water, it's very dilute. And in any little drop, in any little test tube, there'll only be one DNA molecule. It's now purified. I could do that for every molecule there is. The problem is, it's not very useful. Unless I have a way to take that molecule and make extra copies of it. But if I could do that, dilution is purification. The trick we use initially for making more copies is we ask E. coli to do it for us. That, as usual, is the solution to most issues, is find something in life that already does it. And so, just to remember what we did, we took total DNA. Maybe it was yeast DNA. Maybe it was human DNA. Maybe it was zebra DNA. And we cut it up with some restriction enzyme. Okay? Our restriction enzyme we used was EcoR1, and it therefore cut at the EcoR1 sites, GAATTC. We could have used a different restriction enzyme. We then added it to a vector. The vector was cut open at an EcoR1 site. The vector had an origin of replication, ORI, and the vector had a resistance marker, some resistance gene that made a protein that could break down some antibiotic found in nature. We combined these two pieces. Vector now gets its insert. We call this vector. We call this insert. We attach them together using what? Ligase, the enzyme that ligates DNA. We then take this, we transform it into a bacterium. The scale is obviously off, right? This DNA plasmid here is tiny compared to this bacteria, but if I draw it to scale, it won't be very helpful. So we then transform it in here. We do that in a test tube. We treat the bacteria a little roughly so it likes to slurp up the DNA. We then plate it on a plate, and those bacteria that have acquired our plasmid containing the antibiotic resistance gene are able to grow on this plate that has the antibiotic on it. And we call this thing a library. So that's it. We can make a library, and we have an effect on what I said. We've diluted the individual molecules out, and each one went into its own bacteria, and each one got replicated by that bacteria. Those plasmids are replicated by the bacteria. In fact, we choose plasmids that are called multi-copy plasmids, where there's not just one copy, but the cell might make 50 copies of it. We grow up a whole colony of it, and there you go. We talked about some of the issues with it. Where do we get the restriction enzymes, the ligases, the vectors, etc.? It's in the catalog, right? We used to purify them ourselves, but they're all in the catalog. So, any questions about this? We had some questions. I have a question about this. How come when we add ligase, the vector itself just doesn't close up into a circle without an insert? It might. What would happen then? You just have your original vector, and what happens when I transform it into the bacteria? They'll still survive. So will the unimolecular closure of that circle?
Remember we talked about intermolecular reactions and intramolecular reactions, right? And we talked about how alkaline phosphatase could be used so that the vector cannot religate back on itself, right? Removes the phosphates, so it cannot religate back on itself. By and large, you could drive the intermolecular reaction by having a vast excess of your insert and having a fairly low concentration of your vector. Okay, so the probability of a vector and an insert getting ligated is much higher compared to a vector folding back on itself.
Be more common than the bimolecular interaction between a vector and an insert? Probably, because the two ends of the circle are pretty close to each other. So what do I do? Yep. Ooh, that's a cute trick. Two different restriction enzymes.
That's a beautiful trick.
So that they couldn't reclose.
Yes. This also
But then my fragments better have those two different sites too, and I'll only be able to clone those fragments that have the two different ones. But that could work. So you want a trick for making sure it doesn't reclose. Any other tricks? But the problem is I won't get all the fragments, only the ones that have, say, an echo and a BAM site. But that's, so I bring this up not because it's particularly important, but to tell you the kinds of engineering that really does have to go on in molecular biology. What happens is when you have your DNA put in here, we have a sugar phosphate backbone in both cases. And if we look up close, one of these sides has the phosphate, the other has a hydroxyl. Phosphate, hydroxyl, right? And ligase comes in and joins that. Suppose, so this guy has a phosphate on this strand, and this guy has a phosphate on that strand, and hydroxyl there. What would happen if I got rid of the phosphates on the vector?
Do you see what he's driving towards? The use of phosphatase.
So if there were an enzyme that removed phosphates, I could treat my vector first with the phosphate removing enzyme, and now it couldn't possibly reclose on itself. And is there such an enzyme? And what's it called? Phosphatase, because it takes off phosphates. It's all so easy. Phosphatase, it takes off the phosphates. And then it can't reclose anymore. And where do we get phosphatase? It's in the catalog, exactly. So now what happens is that the vector only has an OH here. What happens to ligase? When I put an insert in here, ligase can make a covalent join on this strand, but it can't actually make a covalent join on the other strand. But does it matter? No, because I've closed up my circle on that strand, and I close up the circle there on the other strand, and I just throw it into E. coli. And you know what happens when it goes into E. coli? It's got a nick, obviously, on that strand. It hasn't closed up. But what does E. coli do when it sees that DNA? Must be damaged DNA. I'll fix it. So E. coli actually does the last little trick of closing that up for you with its own enzymes for repairing its own DNA. Now, I bring this up not because it's crucial that you should worry about it, but because I want you to know that there's a whole layer of these interesting engineering tricks that get developed. Every one of them exploits enzymes that we know. Every one of them deals with questions like, will my vector reclose on itself? How do I avoid that? And there's a vast cooking book of protocols in molecular biology, and we constantly are just cribbing from things life does to make our protocols more and more efficient. So I bring it up more because it's kind of a cool thing that all that goes on, and because it helps you remember that these phosphates are very important to joining things up. That's a digression. Now, let's go to the topic. How do we actually read the library? How do we read our library? How do we use the library, read from the library? Well, let's say we're gonna try to find, how about the arginine gene? We talked about the gene for arginine in yeast. So I'd like to clone the gene for ARG1. We found mutants before that were unable to grow without supplemental arginine. They somehow had a defect in producing their own arginine. It's a mutant. I want to find the gene, please. How do I do it? We got to think about what's our insert DNA? What are our vectors? What insert DNA should we start with? Zebra? No. Human? No. How about yeast? We're trying to clone a gene from yeast, right? So let's start with yeast. Okay, so we're going to start with yeast DNA. We're going to cut up yeast DNA. We're going to attach it to our vector. We're going to transform it into E. coli. E. coli will grow up on our plates. And one of these guys, I happen to know it's that one there, contains the ARG1 gene. The problem is I happen to know it, but you don't. How are you going to find out where the ARG1 gene is? Any takers? Yeah. A fluorescent tag. So I should just attach the fluorescent tag to the ARG1 gene. How do I do that? All the DNA looks the same in the tube. How do I know where to attach the fluorescent tag? There's a lot of pieces of DNA there, and my eyes are not that good. Separated. But how will I know which one is ARG1? You see, I don't actually know anything about ARG1. All I know is I made a mutant. The mutant is unable to grow without arginine. I haven't got a clue what that gene is. I don't know what it encodes. I don't know how big it is. I don't know nothing. All I know is that whatever it is, it's a gene which, when mutated, prevents you from growing without arginine. Minimal medium. What if I plate on minimal medium? Now what? What are you hoping for? The one that has the ARG1 gene won't be able to grow. Oh, wait, yeah. But something like that. Let's work it through. We got an idea here. What are we going to do with it? I got a working ARG1. I mutate ARG1 afterwards. Okay, how will that work? I'm open for, you got an idea here? Right? So guys, I have a concern. I'm just transferring this into E. coli. E. coli grows just fine without arginine. I mean, I'm going to take this yeast DNA, I'm going to put it in E. coli. E. coli is kind enough to grow it for me, but frankly, E. coli doesn't need this ARG1 gene. E. coli can grow without arginine. I can plate this with and without arginine. E. coli grows just fine. But you're onto something. You're onto the idea that somehow the only thing we know about ARG1 is that the functional wild-type copy of that gene confers an ability to grow without arginine. And who does it confer it on? And what kind of yeast? Haploid mutant yeast. Ah, so suppose I put a working copy of ARG1, a good copy, a wild type copy, into a mutant yeast. Now what would happen to that mutant yeast? What would happen? The mutant yeast before, could it grow without arginine? No. If I put in a working copy of the ARG1 gene, what will happen? It grows. Now let's design a scheme. Do I want to use E. coli at all? No. What do I want to use? I want to use a yeast. So let's get rid of E. coli, and let's instead use yeast. And which yeast should we use, wild type or mutant? Mutant yeast. What mutation? ARG1 mutant yeast. ARG1 minus yeast. Now, if I plate ARG1 minus yeast on minimal media, what happens? It doesn't grow, dies. What DNA should I be putting in? Yeast DNA, mutant or wild type? Why wild type? Because it'll have a working copy of ARG1. So I want yeast wild type. Now what happens? One of these guys, and only one of these guys here, this one, has the ARG1 gene. That's ARG1. When it goes in, that plasmid has the ARG1 plus gene, whereas other plasmids don't. That cell that inherits that gene there, that gets that gene, is not green. I just drew it green for you. But it has the ARG1 gene. And when I plate this on minimal medium, what's distinctive about it? It grows. That's how you can clone the ARG1 gene. You clone it by the only thing you know about it, namely it confers a function. This is called cloning by function. Or, what did we do when we took wild type and crossed it? What do we do when we cross two mutants together to see if things were in different genes? It was a test of complementation. Really what we're asking is there a plasmid that complements the defect? In effect, what's happening is in this cell right here, we have a yeast cell, and the yeast cell has a defect in its ARG1. But the plasmid has a working ARG1. So for that one gene, this cell could be thought of kind of maybe a little bit like a diploid, just that one gene. And we've done a complementation. Just a teeny little complementation for one gene, and we could call this cloning by complementation. It's essentially cloning by function. Any questions? Yes? Yep. Oh, you see, the yeast genome has about 4,000 different genes. I chop it up with my Echo R1. Some plasmids get ARG1, but most of them get leucine 2 or, SWI6 or other things. And each yeast cell in my library only picks up a plasmid with one chunk of DNA, one gene. So it turns out that the yeast cells in my library, each one has one of thousands of alternative possibilities, and it's just the guy who gets ARG1 who grows. Yes? That's right. Why do I? ARG1 is the ARG1 working copy. In the yeast, I'm talking about this yeast here has the working copy of RH1, RH1 plus. So this guy has an RH1 plus. It also has lots of other genes. Each of these plasmids gets one gene. Some of them get an RH plus. Some of them happen to get a leucine gene that's irrelevant, or they get some other gene that's irrelevant. And the plasmids that contain the working copy of the gene, they, when they go into the cell, give the cell the ability to grow. So that's why. All right, so that's how we get a gene by function.
All right. Okay, there was a call drop and I rejoined. So let's quickly, you saw this lecture, after making a library, the idea is to, look out for genes or fish out genes that we want, right? Mm, okay, so this is something that I thought uh, the previous lecture was like, very interesting, very invigorating. So I thought I should put that first, but ideally we should have done this first. So let's go ahead.
We'll describe what exactly are gene libraries and what are the kind of questions that researchers ask for which gene libraries are needed. We will also discuss the main types of gene libraries and how to construct them, and some very basic techniques for screening of these gene libraries. So gene libraries are also known as gene banks, and essentially they are just a collection of DNA fragments from an organism that represents the entire genome of that organism. So when you make gene libraries, then you have to do a quality check to make sure that it represents almost the entire genome of the organism and you're not missing any part of the genome. And based on the types of starting material that you use, they can be of two different types. Uh, One is the genomic library, which basically is prepared from isolating the entire genomic DNA of any organism. And the second type is the cDNA library, where you isolate the RNA from an organism, convert it into complementary DNA or cDNA, and then prepare the library. So let's now discuss in detail about these two types of libraries. But before we do that, we have to understand what are the questions that require one to create a gene library in the first place. So the first use of the gene library that one can think of is to create a physical maps of all the genes that the organism has. Where are they located in the genome, and what is the context in which they are located? So you might have studied in genetics that the distance between the genes is measured in centimorgan units. Now, those distances were based on the genetic analysis and focused on whether the genes are being segregated together or independent of each other. Based on that, this genetic analysis, one could create the genetic map and say, okay, these two genes are probably close together and these genes are lie far apart in the genome. But to create a more detailed and a more accurate physical map, one needs to start with creating the gene libraries so that you can sequence these fragments and then recreate the entire sequence so that you can study all the elements that are present in the genome. Secondly, if you want to identify genes which are related to a particular cellular function, for example, so you might have studied in cell biology that all the cell cycle genes, CDC genes that were discovered early on in either S. pombe or S. cerevisiae or even Xenopus, they made use of the gene libraries. Similarly, for example, you want to study, for example, how a protein, a secretory protein, is targeted to the endoplasmic reticulum. Obviously, all these functions require participation of many, many genes. So how do you figure out what all gene products are required for a particular cellular function? And you might have studied about the set mutants in yeast, where people used the cDNA expression libraries in yeast to complement the defect in certain functions, and then they can figure out which genes are required for a particular cellular function. Similarly, when you are studying a particular disease process, for example, you are studying cancer, and then you want to compare what are the genes which are expressed aberrantly, or whether there's any new gene expression in the cancerous tissues. So you can create the library from the diseased and normal cells, and then you can do a lot of many different types of tricks to understand the differences between the two type of libraries and the sequences. Disease processes also, sometimes you want to make genome-wide association studies, GWAS studies. So for example, you want to compare whether there are certain single nucleotide polymorphisms in individuals which may be making those individuals prone to develop certain diseases, for example, diabetes or obesities. So for that, you need to do genome-wide study of sequences, compare all those sequences in a population and figure out things. So again, gene libraries are very useful in order to do those kind of studies. Of course, you may be aware that nowadays many sequences from many different organisms have been carried out, and they are all publicly available to researchers. And the starting point for all those genomes to be sequenced is the creation of a good quality genomic library first. Similarly, you may want to do a comparative genomic analysis. You may want to compare the genome sequences of, say, early eukaryotic organism like, say, entamoeba, with something which is, really higher up in the eukaryotic ladder, like a human cell. So you can do a lot of comparative genomic analysis, and for that also you need to create gene libraries first. Of course, the drug discovery platforms, they have made tremendous use of gene libraries, and there are many innovative protocols that have come up in the area of drug discovery using the concepts derived from the gene libraries and DNA sequences. Similarly, if you want to test, for example, the specificity of a particular drug, you have made, for example, a drug against a protein X, but you are not sure whether it binds to any other protein in the genome. So you can take an expression library and you can test your drug against all the proteins that are expressed by that particular cell type or that organism. Similarly, the microbiome research. So you know that many environmental niches, they have very complex, ecological milieu, where there are so many different types of organisms. Now we don't know how to culture all of them. So culture techniques for many organisms are still not there. So what people do, they create a metagenomic library. So they take the environmental sample, they extract the entire DNA from those samples, and then they make what is known as a metagenomic library, so that they can later on understand what are the different types of organisms that are there in that sample, what are the, whether they are, what is the component, gene component, what are the genes that are present, do they have similarity to other things, and so on and so forth. Similarly, our gut flora, is a very complex flora. And so metagenomic libraries and their association to health of an individual is also a very current field of study. So all these very broadly, I have tried to cover different uses of the gene libraries. Of course, there are many different applications.
So this is something that gets asked again and again. So I think make sure you note it down. The uses of gene libraries.
and there are many ways in which the gene libraries have been used and adapted in order to develop more advanced and modern technologies. But right now, we will just consider on very basic aspects of gene libraries. So let's first go over the major steps involved in creation of a gene library. And of course, the first step is to isolate the genomic DNA. Now, most of the genomic DNA isolation protocols are pretty standard protocols, but sometimes there are minor technical details that might differ from one organism to the other. But essentially, you have to first isolate genomic DNA, and then you have to fragment that genomic DNA. Now, this can be done by just sharing the DNA, chromosomal DNA, or you can digest the chromosomal DNA by restriction enzymes so that you have a collection of all these different DNA fragments in a tube. So essentially, the entire genome of the organism is now present in form of these fragments in a single test tube. Now you take a vector which has been, if you use the restriction enzymes for digestion, you can digest the vector DNA with the same restriction enzyme or two enzymes so that the fragments can be cloned into these vectors. If it has been done by shearing, then you can blunt end the fragments and then clone them. Uh, so whichever way you do, you basically then use a DNA ligase to clone all these different fragments into the vector in a single reaction. So you have digested vector in a test tube, you add all the fragments from the genome that you have in that test tube, you use the DNA ligase and let the reaction go. So essentially, in all probability, most of these vectors molecules will have one fragment each that is cloned into them. So then you take these collection of recombinant vectors and then you transform E. coli cells. Now, obviously, because depending on the genome size of an organism and the length of these fragments, there will be a large number of potential E. coli cells which will have which will represent the entire genome of that organism. So let's now look at some of the relevant numbers and think about what are the considerations that you have to deeply think about before you take up the challenging task of construction of a genomic library. So nobody makes a genomic library, very casually. It's not a trivial task. It's a pretty challenging task which requires a lot of technical expertise. And therefore, it is best if you understand at least some of the theoretical considerations before you take up this challenging task. Now, this table here shows the genomic size of various organisms. I have shown E. coli, yeast, S. cerevisiae, Arabidopsis. This is a model plant that is very often used in plant research, and of course the human genome. And as you can see that E. coli genome is about 4.6 megabase pair. And as you go through these different genomes, the size of this genome has increased. But once you come to the Homo sapiens, it is 3.2 gigabase pair genome. What that means is 3.2 into 10 to the power of 9 base pairs. So that is how big a human genome is. So how do you really tackle such large genome to make the libraries, and what are the considerations? So let's first see, supposing you decide to use a restriction enzyme to cut these genomic DNA into fragments. If you use a restriction enzyme whose recognition site is 6 base pair, then the probable frequency of finding such a site in the genome can be given by 1 by 4 to the power of 6, which is once every 4096 base pairs. And this number we arrive at, this kind of calculation can be done, and which assumes that all the 4 bases have an equal distribution in the genome. Then only you will have all the 6 positions at every base, you have the equal probability of 4 different bases to be present. And therefore, this is a theoretical number that we arrive at, that the likelihood of finding a 6 base pair size is once every about 4 kb fragment of the genome. Now, if you use this number and divide the size of the genome by the average fragment length, which in this case is 4096, then the minimum number of clones which will be representing the entire genome of the organism is given here for each of these organisms. So for E. coli, 1,000 clones will represent this entire genome of the E. coli. So this is the minimum number I'm talking about. This number obviously will increase as the genome size increases. And for human cells with the 3.2 gigabase pair genome, the minimum number of clones that you will require to represent the entire genome will be 750,000 clones. So that's a huge number. However, when we do this theoretical consideration, then we have assumed that at all positions, the nucleotides are present in equal, there is an equal probability of all the 4 nucleotides to be present. However, many organisms will not have all the nucleotides present in equal numbers. So there are AT-rich genomes, there are GC-rich genomes, and so on and so forth. So that leads to not having the theoretical calculation to be observed in practice. Similarly, many restriction enzymes, they are methylation sensitive. So if there is a methylation of C residues in the genome, then many sites, even if they are there, they may not be digested by the restriction enzyme that you have chosen if it is sensitive to methylation. And because of these 2 points here, the actual number of fragments that you generate from a genome is quite different from the predicted number of fragments for any given restriction enzyme. So to give you an example, if you take NOT1 enzyme, which is an 8 base pair cutter, then the predicted number of fragments of E. coli genome are 72. However, when you actually digest the E. coli genome by NOT1, the actual number of fragments are only 20. So there can be discrepancy between the theoretical numbers and what you actually get on the bench. So then, which restriction enzyme should we choose? So one consideration that is very important to remember is that we create genomic libraries so that we can sequence them and arrive at the entire genomic sequence of that organism or the sequence of the chromosomes that are in that organism. So in order to do that, because restriction enzymes will cut the genome in many fragments, you want to have the clones in such a way that there is an overlap between the clones, right? So ideally, if you can get one clone to represent one gene, that is the best case scenario. But we know that eukaryotic genes are very, very big. The restriction enzymes will cut indiscriminately between the gene and in anywhere. So you will at least try to achieve to have a specific genomic region to be represented by at least a few overlapping clones so that you can reconstruct that entire genome. So a 4 base pair cutter enzyme is generally preferred, and one of the commonly used enzyme is SOL3A.
Okay. So So we will continue our class on Monday. Any questions so far on this screening part or anything?
No, sir, no questions.
Okay, okay, okay. So we'll start. We'll continue on Monday. Okay. So have a great weekend. Take care. Bye.
Thank you, sir.
Yeah.